Hi dear guys, this is the third lecture of the cell cycle, and in this lecture we are going to talk about the first phase of the mitosis, that is the prophase. So before directly see the prophase, we need to know about few concepts. The first in that is the chromatin. The definition of chromatin: chromatin is a genetic material formed of DNA and histone proteins. We already know that DNA is long elongated structure. Okay, basically in interphase it is thread like structure, and this thread like structure is packed in the nucleus. Okay, it is packed in the nucleus around the histone proteins. So in the figure 2.7 you can see the DNA helix that is shown in the blue, the DNA helix that is wrapped around the histone proteins, and this is the structural and functional unit, that is the nucleosome. Okay, so chromatin is a genetic material formed of the DNA and histone proteins. In that figure you can see the DNA as well as histone proteins. Later on, the second concept, that is the centriole. So centriole is a cylindrical organelle formed of the protein called as tubulin. So in the figure you can see the two centrioles are shown over there. These are the cylindrical structures, and they are formed of the protein, that is the tubulin. These centrioles plays very important role in the cell division. Without centrioles, the cell division cannot be proceed. In the next slide we can see the centrosome. Okay, so centrosomes, these are the structures. The paired centrioles are surrounded by mass of dense material called as centrosome. Okay, that means when the centrioles are surrounded by a mass of dense material, okay, there is mass of dense material surrounding to the centrioles, and this whole structure now called as centrosome. What is the function of the centrosome? Organization of mitotic spindle apparatus. So it plays important role in the organization of the mitotic spindle apparatus. The mitotic spindle apparatus is consisting of the spindle fibers, and in that spindle fibers there are three types of spindle fibers seen. We will see that later on. So what is the spindle apparatus? The cytoskeleton structure formed during the cell division. Okay, the cytoskeletal structure formed during cell division is called as spindle apparatus. Okay, and these spindle apparatus plays important role in the separation of the sister chromatids. So sister chromatids are separated with the help of these spindle apparatus. Next to that, the 3 to 4 concepts. First, sister chromatid. Second, centromere. Third, kinetochore. And fourth, spindle fibers. So in that figure you can see the first figure showing the structure of chromosome, as well as second figure is also showing the structure of chromosome. But what we see in the first figure over there, okay, you can see the two sister chromatids are over there. I have labeled them as chromatid A1 and chromatid A2. You can see the labelings below the chromosome. In red color I have shown chromatid A1, and in blue color I have shown the chromatid A2. And both these chromatids of a chromosome, okay, that is the single chromosome, both these chromatids of a single chromosome, they are referred as, or they are called as, sister chromatids. So what are the sister chromatids? The chromatids of the same chromosome. And you can see both these sister chromatids, or chromatid A1 and A2, they are connected to each other at a point. In the center you can see there is a round structure, and that round structure that holds the two chromatids together, and that round structure is called as centromere. So centromere, with the help of the centromere, the two sister chromatids are attached to each other, or the two sister chromatids are connected to each other. Next to that, the second figure we can see. In the second figure, you can again see there are two sister chromatids, and in the center I have shown that centromere little different than that of the first one. Here you can see centromere is divided into two parts, and each part of the centromere, okay, each part of the centromere on the opposite side, it contains the specialized proteins, and that specialized protein is called as kinetochore. That means kinetochore is the protein that is attached to the centromere on the either side, on both the sides. Okay, that means the sister chromatids, both the sister chromatids on their centromeres, they bears the protein called as the kinetochore, and on this protein there is binding site for the attachment to the spindle fibers. That means spindle fibers, they get bind to the kinetochore, and at the time of anaphase we know that these spindle fiber, they pulls these sister chromatids towards the opposite pole, and finally these sister chromatids get separated from each other. So these two figures, they shows the four concepts: sister chromatids, centromere, kinetochore, and spindle fibers. In next figure, we will start the first phase of the mitosis, which is called as prophase. So before we see the points of prophase, or what events takes place in the prophase, we need to know about the two phases that comes under the prophase: early prophase and late prophase. So this figure shows early prophase, and in this figure you see or what we can see in this figure, you will see the spindle fibers are shown over here. Okay, these spindle fibers, before we know the spindle fibers, just see the centrioles. The centrioles are just going away from each other. In the interface, these centrioles in the cytoplasm, they are placed close to each other, but as the cell enters into the prophase, the centrioles, they gradually moves away from each other. From these centrioles, the spindle fibers are arised. You can see in this figure, from both the centrioles, one centriole, the two centrioles are shown on the upper side, and the two are shown on the lower side. So from these centrioles, the spindle fibers are arised, and these are the spindle fibers which are going to attached to the kinetochore proteins on the each centromere of the sister chromatids. Okay, the second thing we can see in this figure, the chromosomes in the nucleus, they are going to, or they are starting the condensation process. Okay, and another event that you can see, nucleolus is starts to disappear. Okay, or it disappears, we can say. And the next figure, that is the late prophase. In this figure we can see the centrioles are moves very away from each other. Okay, they are now far from each other, trying to move towards the opposite poles of the cell, and the chromosomes becomes condensed. Here we can see chromosomes are condensed, not fully condensed, but they are condensed, and the condensation of chromosome is brought about by the protein, and the name of that protein is condensins. So condensins are the proteins which brought about the condensation of the chromosomes. That means the thread-like chromosomes becomes thick due to the condensin proteins. So, and in this figure of late prophase, you can see the nucleolus is completely disappeared, and with this many cell organelles also start to disappears. In next slide we can see certain points of the prophase. First point is given condensation of chromosomes. As we have seen in the figures, the chromosome starts to get condensed with the help of the protein called as condensin. Okay, the chromosome becomes thick. Next point, each chromosome consists of two sister chromatids. Just we have seen The two sister chromatids are present over there, and both these sister chromatids are interconnected to each other by centromere, we have seen. And these are the daughter DNA molecules. Okay, that means each sister chromatid is the daughter DNA molecule which is produced during the S phase. We already have seen in the S phase the duplication of the DNA takes place. Okay, and that duplicated DNA is nothing but it's the sister chromatid, and in this way the duplicated DNA is interconnected by the centromere. Next point, DNA untangles or separate during the process of chromatin condensation. Okay, that means when the condensation of the chromosome takes place or taking place, at that time the DNA is separating or that DNA is untangled. Untangled means separation of the DNA. Next slide. The condensed sister chromatids are held together by centromere. We have seen that. And centromere is nothing but it's the sequence of the DNA. Okay, the centromere that we see, the two sister chromatids are attached to each other or connected to each other by the centromere, and that centromere is the sequence of DNA. And in the second slide we have seen on the centromeres the protein is found or the protein is bound, which is called as kinetochore. Okay, and to that kinetochore protein spindle microtubules or spindle fibers are get attached. Okay, and from the centrioles the spindle fibers arises. So in this phase we can see spindle fibers are developed or spindle fibers are arised, if you see that figure. Okay, next slide. Centrosomes separate and moves to the opposite poles of the nucleus. That event is already we have seen. Centrosome separates and they moves towards the opposite side. This event takes place in the prophase. In case of higher eukaryotes, we see at the end of the prophase the nuclear membrane get broken down. Remember this point. Nuclear membrane is broken down in the higher eukaryotes at the end of the prophase, and in those organisms in which the nuclear membrane is get broken down at the end of prophase, this type of mitosis is called as open mitosis, where the nuclear membrane is get broken down. But if we see the certain examples like the unicellular eukaryotes or the yeast, in them the nuclear membrane do not break. Okay, as in case of higher eukaryotes the nuclear membrane get broken down, but in case of yeast and certain other unicellular eukaryotes the nuclear membrane do not breaks, and this type of mitosis is called as closed mitosis. The nuclear membrane is not breaking, so the chromosomes are inside the nucleus. Okay, so this is the closed mitosis. Next slide shows the main events that takes place in the prophase. So what are the main events? First event we have seen condensation of the chromosomes. This is the main event in this prophase. Second, we have seen on centromere the protein is bounded, that is called as the kinetochore. Okay, and to this kinetochore the spindle microtubules are bind. So this event takes place in the prophase. To the kinetochore spindle microtubules get bound. Mitotic spindle fibers are developed, that is we have seen, because after developed they get bind to the kinetochore. So the point 4 should be first and point 3rd second should be later on. And fifth point shows us that centrosome separates and moves to the opposite side of the nucleus. The centrosomes they get separate and they move towards the opposite sides of the nucleus. Okay, so at the end of prophase we have seen the centrosomes are completely moved towards the opposite poles. Okay, and the chromosomes becomes condensed, the nucleolus disappears, and some of the cell organelles also disappears. Now we are going to see the next phase, that is the metaphase. Metaphase is the second phase in the mitosis. We can see in this figure the chromosomes are arranged in the center of a cell. Okay, center of a cell, and that center of cell is called as a metaphase plate or equatorial plate. Okay, And in this figure we can see the centrioles or the centrosomes, they are completely on the opposite side, and from these centrioles or centrosomes the spindle fibers are arises, and these spindle fibers are get connected to the kinetochore proteins on the each sister chromatids on both the sides. Okay, so we'll see the next slide. What events takes place in the metaphase? First, the nuclear membrane is completely degenerated. Okay, the nuclear membrane is completely degenerated. That means the structure that you can see in the last figure, that was completely in the cytoplasm, because there is no nuclear membrane. Okay. Now chromosomes, centrioles, spindle fibers, all these things are in the cytoplasm. Second point, chromosomes spread in the cytoplasm of the cell, as I told just right now. The condensation of the chromosomes is completed. Okay, chromosomes are condensed now, and the chromosomes seen as very short, very distinctly under the microscope. So as the chromosomes become very short and they becomes very thick, so we can see them under the microscope very clearly. So actually the metaphase can be very clearly seen and observed under the microscope, and we can identify it very easily because the chromosomes are arranged on the equatorial plate. Okay, so here the metaphase chromosomes consisting of the two sister chromatids, as we can see in the figure. The chromosomes, they shows the two sister chromatids, and the chromosomes are arranged on the equatorial plate. You can see in prophase you have seen the chromosomes were here and there in the cell, but here in this metaphase the chromosomes are arranged on in the center of a cell in the equatorial plate, and that equatorial plate is called as the metaphasic plate or metaphase plate. This is the imaginary line. Okay, and next point, the spindle fibers are fully formed. We can see in the figure the spindle fibers are fully formed and they attaches to the kinetochore proteins on the centromeres. As now the chromosomes are arranged on the equatorial plate, and the cell can be ready to further proceed into the anaphase. Okay, but it is very important to note that before the cell enter into the anaphase from the metaphase, all the chromosomes must be arranged on the equatorial plate. This is very important. If there is any single chromosome to which the kinetochore proteins are not to the kinetochore proteins the spindle fibers are not attached, okay, and if that single chromosome is left to get arranged on the equatorial plate, then this type of cell will not be enter into the anaphase, because what is happening in the anaphase the chromosomes are going to sister chromatids are going to separated from each other. That means the equal distribution of the sister chromatid is going to take place in the anaphase, and if in the metaphase any single chromosome it fails to get attached to the spindle fibers, then it will not goes into the anaphase. So if it doesn't go into the anaphase, that means there is abnormality in the number of the chromosomes in the anaphase. So in a normal condition this doesn't happens. So particularly cell waits metaphasic cell waits until all the chromosomes in the metaphase cell get all the kinetochores in the metaphasic cell must be get attached to the spindle fibers. Okay, then and then only the metaphasic cell can enter into the anaphase. And if that cell is not ready to go into the anaphase due to the not attachment of single or many chromosomes by the spindle fibers, this cell will rest into the metaphase for a particular time period until that single or both the chromosomes get attached with the spindle fibers, and after binding with the spindle fibers, then and then only that cell can enter into the next stage, that is called as the anaphase. So in next lecture we are going to see about the anaphase. So till that, goodbye. Don't forget to like the video and subscribe my channel.